I am Aliasger Salem, Ali for short, and I'm from the University of Iowa. I have known Phil for a long time, many, many years. We've had him visit the University of Iowa, and we've been collaborators for many years. And so it's a great pleasure to be able to have spent the time with him during these last few days at Harvard, and talking about potential collaborative opportunities and discussing science, and meeting with many of you, and I'm very pleased to see you here as well. Now today I'm going to talk about the effect of inhalation exposure to metal oxide nanoparticles and the efficacy of that, or how that affects the efficacy of a vaccine against house dust mite allergies. And the structure of the presentation is that I will start by talking about the development of the vaccine itself, how it came about, how we started the formulation of the vaccine, how it works in a murine model, and then from there I'll talk about the toxicity of copper oxide nanoparticles, and then the last portion to close off the overall presentation will be the impact of the copper oxide nanoparticles following an inhalation and how that affects the efficacy of the dust mite allergy vaccine that I am going to be talking about, having developed at the start of this presentation. And so because of that format, you'll see some old data to start with, some more recent data as we get towards the middle of the presentation, and then some unpublished data towards the end. Okay, so to start with, I'd like to talk about the preparation of a dust mite allergy vaccine and how we developed this concept and how we prepared it. And the core ingredient to developing, or the core ingredients to developing this vaccine are CpG Oligonucleotides, which act as an adjuvant, the antigen, which we are stimulating an immune response against, and then a delivery system that packages both the adjuvant and the antigen into a single delivery system to generate a robust antigen-specific immune response. As part of that overall package, the delivery system, I'll talk about the choices of antigens and how we identified them. I'll talk about the mechanism of action, and I'll also talk about the formulation. And in the spirit of this being a nano lecture, I will also talk about the impact of the size of the particles used in this vaccine formulation and how that impacts the overall immune response to the antigen. So, CpG oligonucleotides. I believe I need to use the mouse pad for this. They are these short single-stranded DNA molecules that contain a cytosine followed by a guanine, and then they have a modified phosphorothioate backbone. CpG was originally identified from bacterial DNA to be a very potent antigen-specific, or a very potent immunostimulatory sequence. And when someone has a bacterial infection, it stimulates a very robust immune response against it, and that's why bacterial infections can be cleared by themselves. And a key sequence in bacterial DNA that causes that immune response to be effective and to be able to clear away the bacterial infection is these CpG sequences. And there were two individuals that many, many years ago identified the CpG sequence and were able to commercialize it and turn it into an adjuvant product that could actually be used in the clinic. One of those was Akira in Japan, and the other one was Art Krieg at the University of Iowa, where after patenting the sequence and developing it as an adjuvant, he formed a company called Colege Pharmaceuticals, and Colege Pharmaceuticals was then bought out by Pfizer, and this was an adjuvant that had tremendous interest across the pharmaceutical industry for its potential applications in vaccines. Now, from a delivery perspective, the CpG can be used it can be packaged in microparticles, it can be packaged in liposomes. You can actually form CpG antigen conjugate molecules, and there were some products based on this idea from Dynavax, for an example, that were very, very potent vaccines against a number of diseases. And when you utilize CpG along with a specific antigen, the impact of that is that you can have upregulation of IL-12, TNF-alpha, interferon gamma. So these are all pro-inflammatory cytokines. And you can have upregulation of interferon gamma from NK cells, which can have an impact on improving cancer therapy. You can potentially reduce allergic and asthmatic responses because of the modulation of the immune response as a result of using the CpG. And so there are a lot of potential applications in vaccine settings where CpG can be a really powerful adjuvant. Now, to put this in the context of how adjuvants might be used in other settings, when most people get a flu vaccine, it will be an adjuvanted vaccine where the adjuvant is alum, and that's the most common FDA-approved adjuvant that's used in most vaccine settings, but it's not very effective, and it doesn't do a very good job of giving you these pro-inflammatory cytokines and this Th1 type response that is considered very therapeutic relative to how effective CpG is. So that's the first ingredient to our vaccine formulation and the first thing that I wanted to talk about. Now, Metsitov, Ruslan was his first name, identified in a paper that he had published in Nature, and this was in 2006, that the efficiency of presenting antigens from phagocytosed cargo is dependent on the presence of TLR ligands within the cargo. So now I'll explain what this sentence means. A TLR ligand is a toll-like receptor ligand. CpG is a ligand for toll-like receptor 9, which is found in the endosomal or phagosomal compartments of dendritic cells and macrophages. And when CpG binds to TLR9 via a MyD88 pathway, if in combination with an antigen, it can help with class 1 processing of that antigen and give you class 1 presentation of the antigen, which then can go on to stimulate very strong CD8 positive T cell responses against the antigen. And in this statement, where Ruslan Metsitov has identified the criticality of having antigens and the TLR ligands together in the same system, in the same compartment, this provided me with the opportunity to set up a research program on delivery systems that could co-deliver both the adjuvant and the antigen. Because the one nice thing about this Nature paper was that it identified the problem but did not propose the solution, which really gave an opportunity for someone like myself focused on delivery to come in and address that issue. So conceptually, the general approach of how we would take this vaccine and how we would build it is that you would take a biodegradable microparticle. The microparticle would be made out of something like polylactic co-glycolic acid, which is PLGA. PLGA is used in a wide range of biomedical applications, from depot-based delivery systems to things like sutures, where if you have a site that needs to be stitched up, if it's stitched up using this PLGA type material, over time the suture will degrade into glycolic acid and lactic acid, and then via Krebs cycle into carbon dioxide and water, so that the suture resorbs away naturally, and all you have left is the repaired site. And so many people, I don't know how many people have had suturing done to them and have not had to go back for a second procedure to remove the suture, but the reason you have not had to go back is because it's resorbable and it breaks down into natural products. So we use that same material, the PLGA, to form a microparticle. And the microparticle has the antigen and the adjuvant entrapped within that particle with the polymer matrix, and it's a physical entrapment, so there's no chemical modification to have the antigen and the adjuvant inside this particulate matrix. And over time, as the particle degrades and through diffusion, the CpG and the antigen is released, but because it's a particulate, when the particle is injected, it's seen as debris, foreign debris, and it's in a particular size range, so that as it's seen as debris, you have macrophages, dendritic cells, all the kinds of things that will want to come and clear debris. They migrate in. They see this particulate debris, and as the particle is interacting with the antigen-presenting cell, the macrophage, then the arms of the macrophage will come. It will engulf the particle, it will bring it down, and then invaginate away from the surface, forming an endosomal compartment or a phagosomal compartment. And that compartment is where the toll-like receptor 9 receptor for the CpG exists. So you have a very targeted mechanism to have a particle delivering your antigen and your adjuvant for that receptor at the target site through this delivery mechanism. And once the particle delivers it within the acidic environment of that phagosome, the CpG is released, the antigen is released, you get class 1 processing of that antigen via the MyD88 pathway due to the CpG, you get class 1 presentation of the antigen. That then migrates to the lymph nodes and stimulates naive T cells to become cytotoxic T lymphocytes that are specifically responding or engineered to respond to the antigen that was originally loaded in the particle vaccine. So that's conceptually how this particle vaccine has been developed. Now, in order to prepare the vaccine, I'm a pharmaceutical scientist, a formulation scientist. We are particularly interested in the formulation parameters and how that impacts the properties of the particle vaccine. And so we have shown that by controlling the stirring rate and the surfactant concentration, you can control the size of the particle vaccine, where the higher the surfactant concentration and the higher the stirring rate, the smaller the particles. So you've got the highest surfactant concentrations here at 9%, and you've got a high stirring rate. You're preparing 200 nanometer size particles that are loaded with your antigen and your adjuvant. If you take 0.1% and the lowest stirring rate, you've got 10 micron particles. And then you have an intermediate series of values from 10 microns all the way to 200 nanometers that can be engineered by the formulation parameters that you use, including stirring rate and surfactant concentration. And to give you a conceptual idea of how these particles form, if you have an oil and you add it to the water and the water is rapidly stirring, you'll form an emulsion because oil and water are immiscible. And the dichloromethane in a system like this is completely immiscible with water. So our dichloromethane will have our polymer and our antigen and adjuvant, and then you place that into this rapidly stirring water phase, which is completely immiscible with the dichloromethane. You form lots of droplets of these dichloromethanes that are immiscible, and they are stabilized by the surfactant, which is partially hydrolyzed, so it's got hydrophobic and hydrophilic properties, where the hydrophobic component of the surfactant associates with the dichloromethane, and the hydrophilic component associates with the water, stabilizing those droplets with the polymer and the antigen and adjuvant entrapped within that matrix. And then, because the dichloromethane has this, it's a volatile solvent, and so when you have it stirring over a period of time, over hours, the dichloromethane begins to evaporate off, and you're left with the hardened polymer particles in the stirring solution. And so that's how you form these particulates. And the controlling factor that allows you to control the size is the surfactant concentration, the concentration of the polymer and the loaded agent, and then the stirring rate. So a variety of parameters to really control the size that you're interested in. Now, the surface morphology of these particles are smooth and spherical in appearance. This matrix is a polymer matrix loaded. It's PLGA, as I mentioned. It's a safe, biocompatible polymer. And your CpG and antigen is loaded throughout that matrix. And when you look at the release profiles of the loaded agents, you see that you get a burst release followed by a more sustained release over time. Now, the reason that you have this type of profile is because over the protein and CpG are very hydrophilic. And so because they have a high degree of hydrophilicity, you can imagine that when you form an oil in water emulsion, that as the particles are forming, a large proportion of your hydrophilic material is going to orientate itself towards the surface, as well as giving you some that is loaded within the matrix. And that surface immobilized or surface, near surface loaded proteins and adjuvants releases very quickly, where you get almost 30% of the loaded properties or the loaded agents releasing within the first 24 hours, followed by a more sustained release over time. Now, in the setting of a vaccine, that is particularly advantageous because you want to stimulate a robust immune response initially, followed by the prospect of maintaining that response over time. And so this release profile matches greatly with our desired delivery profiles to get a robust response and then maintain it. Now, once you've made the particles and you've shown that they work, the next thing that you want to be able to do is to show that it actually has activity in an in vivo model. And so we injected CpG and antigen into mice, both in solution and in a microparticle formulation, and then we had a variety of controls, including over in the microparticles alone, and then over by itself. And over here represents ovalbumin, which is a xenogeneic model antigen that can allow you to determine the differences between the various groups. Now, what you can see here is that if you deliver antigen alone, the antigen alone does give you a modest response that you can't detect. But more importantly, when you inject CpG, this adjuvant, with the antigen, you see a fourfold elevation in the antibodies against that antigen. It's really clear that the presence of the adjuvant is enhancing the response. But now take a look at when you load the CpG and the antigen into a microparticle formulation, and what you can see is that as you have moved from just putting the CpG and antigen in solution here, as you move it into a microparticle formulation, you see a significant and robust enhancement in the antigen-specific immune response that really clearly illustrates this concept that co-delivering the CpG and the antigen in a microparticle formulation significantly enhances the antigen-specific immune response. In addition, it's worth pointing out that the CpG and the antigen loaded in the microparticles was a lower concentration than the CpG antigen just delivered in solution, suggesting that in addition to enhancing the response, there is also a dose-sparing effect as well. So this formulation strategy, this general approach, really could enhance the response significantly. When we looked at whether these microparticles could improve pro-inflammatory markers and activation of dendritic cells, we saw via CD80 and CD86 that these microparticles were very efficient at triggering activation and maturation of dendritic cells. So in addition to the delivery attributes, you also have the ability to use the microparticles to stimulate activation of dendritic cells. And when you look at the pro-inflammatory cytokine, interferon gamma, which is often associated with a T helper 1 type immune response, the key data here that is really, really significant is that when you take over and CpG and you inject that, or you incubate the DCs with just that alone, you see a 37 value of picograms per ml of the interferon gamma. Take the over CpG and place it into a microparticle formulation, and you see a 20-fold enhancement in the secretion of interferon gamma, that pro-inflammatory cytokine, suggesting that, again, in addition to the antibody responses that you're seeing, the microparticle formulation significantly enhances the pro-inflammatory properties of the delivery system and really enhances secretion of that in a way that we expect will enhance the antigen-specific immune response that we're trying to trigger, which is focused on a T helper 1 type immune response. In addition, in a study that I did with Anthony Sandler, we took tumor lysate and we packaged that with CpG into a microparticle, and then we compared that to just tumor lysate alone in microparticles. And what we found is that with the various controls, microparticles loaded with a broad spectrum of tumor-associated and tumor-specific antigens that come from the tumor lysate plus CpG, you get the same thing we saw in the previous slide, which is this upregulation of these pro-inflammatory markers of interferon gamma, but you don't get upregulation of immunosuppressive cytokines like IL-10, suggesting that you're getting a really good portfolio of pro-inflammatory cytokines without the undesirable suppressive cytokines, suggesting that this is a really good formulation vaccine strategy. Now, we also wanted to look at the subtypes of antibodies. So I mentioned that in a vaccine strategy where you're trying to get a T helper 1 type immune response that is associated with positive outcomes in treating infectious diseases or allergies, you want to get a T helper 1 type response. And a T helper type 2 response is less desirable and will often exacerbate those allergic responses. And the way to characterize which type of response you're getting is to look at the subtype of the antibodies. So an IgG1 response is more typically associated with a T helper 2 type response, which is not our target, a desirable response. And an IgG2A or 2C antibody response is associated with a T helper 1 type immune response, which is our target type of response. And so if we focus in on the IgG2A response, which is the one that we're most interested in, if you package the CpG and the antigen in a microparticle, you see very, very robust IgG2A antibody responses, suggesting that you're getting a robust T helper 1 antigen-specific type immune response. If you take CpG alone or over alone, you can see that the T helper 1 type immune response, the IgG2A antibody response, is significantly muted compared to when you package both of them into the particles, so that you're getting this much stronger IgG2A antibody response. In addition, another important control that is presented in this study is that you can see that when you package the model antigen ovalbumin into a microparticle and you add soluble CpG, so you're not putting the two together now, even though you're delivering both of them, the response, again, for the IgG2A antibody response is significantly muted relative to putting them together, suggesting that the co-delivery hypothesis of needing to have both the antigen and the CpG in the same cargo is confirmed using our antibody response measurements. So now I'm going to talk about a parameter that I expect that a significant proportion of this audience here is going to be excited about, and that is the idea that size matters. And this is a nanolecture, so I think that there will be a significant agreement with this broad concept. Now, in the vaccine delivery systems that we prepared, the first thing I showed you was that based on the formulation characteristics, you can control the size of the particles. And that proves really valuable for investigating the impact of the particle size on the magnitude of the immune response that you stimulate. And so what we do is we prepare a wide range of particle sizes, and using differential centrifugation, we can have good uniformity with reduced polydispersity, and we can have clearly defined size ranges going from 16 micron size particles to 7 micron size particles to 1 micron size particles to 200 nanometers. So you can see that you can create a broad range of sizes where we can test the potential responses based on the size of the particle vaccine. And what I'm going to show you now is that the size of the particle that you inject will affect the magnitude of the immune response. And I'll show that on the next slide by showing first how size impacts uptake. Now, the results here show that if you have a large 17 micron size particle and you incubate them in macrophages or in antigen presenting cells, those macrophages have a really hard time internalizing the particle. And you can imagine why, because these particles are almost the same size as the cells. So trying to internalize that thing is really going to stress the cell out. And so you don't get great uptake of 16, 17 micron size particles. But as you decrease the size from 7 microns to 1 micron to 200 nanometers, you can see, based on the confocal images, that there is a proportional decrease, or there is a proportional increase in the uptake of the antigen-presenting cells as the particle gets smaller. So this is why the need to characterize nanoparticles and how they create toxicity, but also inflammatory responses matters so much, because the size of the particle dictates how effectively they are taken up by antigen-presenting cells, with the smaller particles being taken up much more effectively than the larger particles. Now, that result through confocal, which we verified is not just surface associated, but is also internalized using Z-stacks, we can then reconfirm using flow cytometry, where once again, you can see that if you're looking at uptake using flow cytometry, the largest particles have the lowest uptake, and the smallest particles, these 300 nanometers, have the greatest uptake. And it makes sense. The smaller the particle, the more likely it's going to be effectively taken up by cells. And there is a proportional relationship between size of particles and uptake by your target cell population. And so this reiterates the same results that you saw qualitatively in the previous slide, that there is a size relationship to uptake by cells. Now, in terms of vaccine efficacy, you need your particles to be taken up and to be activating your dendritic cells and macrophages and antigen-presenting cells, because that is the target cell population for stimulating an immune response. And we mentioned that one marker of activation of dendritic cells is CD86. And what you can see, again, is that take a look at this beautiful profile where the largest particles give you the lowest activation, and as you increase, as you see increased activation of the dendritic cells, you can see that's proportional with the size of the particles, where the smaller the particle, the greater the activation of CD86, and that's consistent with this idea that you're delivering your active agents to the target sites within the dendritic cells and within the artificial antigen present, or within the antigen presenting cells, the professional antigen presenting cells, of which the dendritic cells are one of those. So there's a proportional relationship. Now, one question that comes up is, how do you know that it's not just to do with release? Because the smaller particles have a greater surface area by mass. You'll get a much, much quicker release of your antigen and your CpG then if you're in a smaller particle than if you're in a larger particle, where the path to delivery is longer, for release is longer. But what you can see here is that in the solution, or the soluble CpG and antigen, where you have immediate release of all of your agents, you're not getting as strong an activation of the dendritic cells and macrophages as you do when you have that 300 nanometer size particle. So it's not just to do with release, but it is a size-dependent effect as well. And the other thing that I should address right now is that since I'm showing that 300 nanometer size particles are so much better than 17, 7, and 1, why not keep going smaller? Okay, that should be the immediate question that comes to all of your minds. And I know that as I'm seeing smiles in the audience, you are thinking that. Now, the reason is to do with loading. So if you go much lower than 200 or 300 nanometers, you compromise your capacity to load this CpG oligonucleotide and this large protein into the particle, and so you have a threshold where you can no longer go any smaller, because you would reduce your ability to load the active agents that are required to stimulate a response. And so that is the reason that is the cutoff threshold for why we did not go any smaller. And in any nanomedicine and drug delivery type project, the size of the particle is a parameter, but your capacity to deliver therapeutic load is also a critical parameter that you must consider in your formulation. Okay, so how does the size of the particle affect the magnitude of the immune response? You can see that 17 micron particles don't give you a very strong antibody response. Here is the experimental design: day 0, day 7 injections, antibody titers at day 28, and then CD8 positive T cells being measured at day 14 and day 21. to determine whether we're getting that population of cells that we're really interested in. So here you can see 300 nanometer particles give you the largest antibody response relative to 1 micron, 7 microns, and 17 microns. And when you look at the ratio of the IgG2A to IgG1, you can see again that the smaller the particle, the stronger the T helper 1 type immune response relative to smaller particle or relative to the larger particles. And it is a proportional increase where as the particle gets smaller, the T helper 1 type immune response, as measured by the ratio of the IgG2A to IgG1, gets bigger. So again, you see that proportional increase. And then when looking at CD8 positive T cell responses, which are critical for treating cancer, which is another major emphasis of my program, you can see again that the smaller the particles, that for going from 17 to 7 to 1 to 300, the greater the CD8 positive T cell response, suggesting that there is a proportional increase in the response as the size of the particle gets smaller. Now, in the next slide, I'm going to move on to how this could be used in a dust mite allergy vaccine. But in the AAPS journal, in Cancer Immunology Immunotherapy, in the Journal of Immunotherapy, I've published a series of studies that show that these particle vaccines, when loaded with tumor-specific antigens, can be used to stimulate a CD8 positive T cell response against those antigens, and we have responses against lymphoma, breast cancer, melanoma, and we have shown that this particle-based vaccine strategy can really be used against a broad range of cancers to stimulate immune responses that reduces and suppresses that tumor growth. In addition, we've also shown that when you combine them with checkpoint blockade agents of the like that were discovered by James Allison, the recent Nobel Prize winner for the anti-PD-1 antibodies, you can really, really effectively suppress and treat cancer using a combined vaccine and checkpoint blockade agent-based approach. And one of the papers that really describes that very well, again, is in the AAPS journal. Now, I want to focus in on the dust mite allergy vaccine, though, because as you look at this dust mite, this creature produces excrement, and that excrement has allergens that causes a very robust allergic response in a wide range of patients. Now, in collaboration with Peter Thorne, who is my main collaborator on the development of this dust mite allergy vaccine, he is also one of the leads on the UO1 consortium together with Phil to look at nanotoxicity. We were able to develop a particle-based vaccine that could stimulate an immune response against a dust mite allergy. And it built on this early studies looking at co-delivery of a model antigen and adjuvant. And so we wanted to use the same conceptual approach to stimulate a response against dust mite allergies. And again, Peter Thorne was my collaborator. Andrea, who is also on the UO1, participated in this, and then myself as well. So dust mite allergies, what are they? It's an overreaction of the immune system to an allergen. 84% of US residencies have detectable levels of house dust mite allergens, and a quarter of these houses have higher levels of allergens than the proposed limit for asthma. So you can imagine that's a fifth of the entire US nation, and it's not just the US, but a fifth of the entire US nation has higher levels of dust mite allergens than the proposed limits for asthma. So it's a really prevalent problem. And DERP1 and DERP2 allow for diagnosis of more than 95% of the patients. So that's a really good target antigen to try and stimulate an immune response against. And this chronic exposure to these allergens leads to lung eosinophilia, airway obstructions, and asthmatic attacks, and also some of these types of responses that you see in the picture that can really be undesirable in patients, and it's really prevalent. So what we did was, if you look at the current modalities of treatment, you can see that there's mast cell stabilizer, there's beta-2 agonist, neutralizing antibodies, desensitization, which is a subcutaneous immunotherapy. where, to give you an example, Johns Hopkins did a clinical trial over 3 years where they had pediatric patients and they put dust mite allergens under the tongues on a regular basis. And then after 3 years, they showed that they could desensitize to the dust mite allergens. But 3 years is a long time to be able to do this. If you can vaccinate to achieve that and shift the T helper 1 and T helper 2 type balance to alleviate and mitigate those responses to the dust mite allergy, that would be a really big deal. So we decided that we wanted to pursue this approach, and it was particularly relevant to the EHSRC and Peter Thorne's research because it's an environmental exposure that triggers this. So how do we do this? Well, we take a vaccine loaded with dust mite allergen antigen, the DERP2 or DERP1. We immunize. We hopefully will trigger a response that gives you an IgG2A antibody response, which is associated with a T helper 1 type immune response. If we don't, if we get this IgG1 response, IgE and eosinophils, that would likely correspond to a patient having a very robust allergic response. So it's undesirable. So things that I look to as markers for likely to be an undesirable response are IgE and eosinophils, and a positive therapeutic response was likely to be associated with IgG2A, which is what we're trying to achieve, and you saw in the model antigen gave us that type of response. Okay, so bad, good. That's kind of the simplistic approach. So we have our plan here, day 0, day 7, we vaccinate, we give daily exposures of the allergen to the mice, and then we have small, medium, large, with and without CpG. We've got shams, sentinels. We've got all the necessary controls to look at whether we get a good response. This is the study design. And what you can see is, remember what I said about eosinophils not being desirable? We'll take a large particle with just the allergen, the DERP2, and you see this massive eosinophil response. And then the moment you add CpG to the formulation, that drops dramatically. But that's not the only parameter. If you look at the smaller particles with DERP2 and CpG do a really good job of reducing that eosinophil response that is undesirable relative to the large particles without CpG. So the two take-home messages, even on this first slide, is that smaller particles with CpG and the DERP2 will do a great job of mitigating eosinophil responses. And you can see that when you look at the IgG1 response associated with T helper 2 type response, large particles, look at that large response. You take a CpG, it drops significantly. And then if you look at the IgG2A response, the presence of CpG consistently increases your T helper 1 type antibody responses, so the IgG2A. And you can see that consistently. So look at medium particles, add CpG, it goes up. And then look at the ratio. There is a size-dependent relationship, and then the inclusion of CpG consistently makes the vaccine more efficacious against a dust mite allergy. So this is sort of highlighting that key trend that you can see there. Now, when they test for dust mite allergies, one way that they test for the asthmatic response in the clinic is to go for a methacholine challenge. And so one of the things that we did, and again, this is a collaboration with Peter Thorne and Andrea, we looked at the methacholine challenge for mice with small particles, medium particles, large particles vaccinating, and with and without CpG. Now, the large particle with DERP2, we don't expect it to work. And so we expect a terrible methacholine response in terms of the airway resistance. And look at that. If you look at the large particles with just the DERP2 and no CpG, you get a really massive response. Include CpG and look how well that drops. And then you look at the medium to small response, the smallest particles with CpG give you really, really great mitigation of the response to a methacholine challenge because of the particular vaccination strategy. So again, this shows that this is a very, very effective approach for potentially countering dust mite allergies. Now, we also see that if you look at the lung histopathology, and this is to give credit to Catherine Gibson-Corley for the core pathology core for helping us to do the sectioning, you can see that small particles, again, you get a much lower lung inflammation than you do with large particles. And the best way to show this is to look at the quantification on the next slide, where you can see that you get this perivascular cuffing, this perivascular inflammation is the largest with the large particles with DERP2. Add CpG, it drops, and then you can see a consistent reduction in the inflammatory response as you reduce the size of the particle vaccine and you include the CpG. Okay, so now I want to talk about copper oxide nanoparticles and its toxicity. And this is something that's truly well established. The groups here have had a lot of experience with this. Peter Thorne's group has had a lot of experience with this. I have had a lot of experience with this. And broadly speaking, if I was going to summarize what we have identified, together with Vicky Gracian, we've shown that there are size-dependent effects on copper oxide nanotoxicity in lung epithelial cells, where the size of the particle can have an impact on the magnitude of toxicity. We've also showed that coating copper nanoparticles with chitosan might reduce in vitro toxicity, but its mucoadhesive properties will lead to adhesion within the lung, and that causes retention of the copper oxide nanoparticles in the lung area. So that actually increases inflammation. And so that's a discrepancy between in vitro and in vivo responses because of how long the copper oxide nanoparticles are there. But broadly speaking, we know, and it's well understood by this group, but also by a number of other groups, that copper oxide nanoparticles induces toxicity and inflammation. A lot of that is related to the copper ions that from dissolution that are released from the copper oxide that is responsible for that toxicity and that inflammation. And as part of the studies, we have always been very, very interested in carefully characterizing our nanoparticles before we use them. And so the types of things that we would do are TEM, XRD, XPS, looking at the particles by the various methods of characterization. And one of the key things that we determined, even in terms of sizes, is that if you have different sizes, they have different compositions, and you can identify that in XRD. So the work that the ERCC is doing here now to send particles to us for testing in lung inhalation studies is so critical, because we know that it has such a big impact on the overall composition of the materials and how they look. And so we have seen that that has a big impact on overall toxicity. We've also seen, as I mentioned, that the size of the particles can have a really big impact on the magnitude of toxicity, but we've also identified that dissolution of the copper oxide nanoparticles and the copper ions can be a major factor in the response that you get in terms of toxicity. And this is particularly true in vitro, but we've also seen that this has a massive effect of in vivo when a model is exposed to inhalation exposure of the copper oxides. And we have a whole series of publications that have identified this. And then Peter Thorne's group has done a lot of work on lung inhalation of copper oxide nanoparticles that has shown this relationship and really well characterized it. So the question that we had, and this is part of our UO1 proposal that is part of the consortium with Harvard and with our other collaborators at UCLA and other places, is can the copper oxide nanoparticles impair the dust mite allergy vaccine that I just described how we developed in the previous slides? And so that's sort of the project that we're currently working on and that we're developing data for, is we want to know whether what we know has an inflammatory response and a toxic response. Can it impair the dust mite allergy vaccine? And can that be an issue then not only for dust mite allergy vaccines, but also for other vaccines where people in an environment that are exposed to copper oxide nanoparticles have to take this into consideration? So I have a really nice picture of Phil here. I took it from the website. It has a nice background with it as well. And I want to mention the Nanosafety Research Center. We are taking copper oxide nanoparticles and other nanoparticles, metal oxide nanoparticles, a wide range of materials from the ERCC that is headed up by Phil. I want to acknowledge Demetrios and other people here that have helped with the characterization. Also Georgios, who was here previously, but they do a great deal of characterization on those materials, looking at XRD, TEM, size, zeta potential, and they send us these long files of this characterization, and we know that that's important and critical because we've done those type of studies in the past, and we know how important that data is. We take that data, and we take those nanoparticles that are very well characterized, and we're now testing them in an immunotoxicity mouse model where we're looking at the impact on a dust mite allergy vaccine. And the dust mite allergy vaccine I've described very clearly in the previous slide, so I want to know, and this is again a very strong collaboration that involves Sudhatip, a graduate student, Andrea, a research scientist that is really phenomenal in these type of studies, and then Peter Thorne, my longtime collaborator. And we want to know whether that decreases the efficacy of the HDM vaccine. So what we're doing is we're looking at exposure to copper oxide nanoparticles via inhalation over a long period of time. Again, ERCC takes credit for the materials. Then we're doing a subcutaneous injection of the vaccine that I previously described. And then we're doing, after we've done this inhalation exposure and this vaccination, we're looking at nasal instillation of the house dust mite allergen over a prolonged period of time with all the appropriate controls to see whether that makes a difference or not. And in preliminary studies, so this is unpublished, we're just generating these results right now, what you can see is that the copper oxide, when it comes to IL-4, when it comes to IL-6, KC, IL-5, is consistently having an impact on cytokine production relative to using just the dust mite allergy vaccine by itself. So the early results that we are generating at this point, and there's a lot more work that needs to be done, but the early results across a wide range of cytokines in BAL fluid is clearly indicating that inhalation exposure to these copper oxide nanoparticles is impairing vaccine activity. You can also see this again with IL-10 and then interferon gamma and TNF-alpha. If you consistently look at the vaccine response versus copper oxide with the vaccine response, there are clear cytokines, it's not all of them, but the key important cytokines where you see a significant difference as a result of having copper oxide nanoparticles being exposed to the mice prior to vaccination. So our early indications are that the hypothesis is correct, that this kind of exposure can impair vaccine efficacy. Again, let's take a look at differential cells. You've got macrophages, lymphocytes, eosinophils, and it's clear that across a wide range of cell populations, that copper oxide nanoparticles can have an impact on the type of cell balance and cell population of inflammatory cells that you would get if you were just using the dust mite allergy vaccine alone. Now, there's a lot of question marks about this data because we need to make sure that we're repeating it. Some of the other things that we are also needing to do is that we have a plan of action right now that I'll hopefully show on the last slide that is looking at what happens to the inflammatory cell populations at different concentrations of copper oxide nanoparticles in terms of inhalation exposure and different concentrations of the dust mite allergy vaccine, so that before we redo many of these experiments, we really have finessed and fine-tuned and optimized how much exposure of copper oxide nanoparticles we should do, how much of the vaccination we should provide, what is the overall dose that we should be providing, so that we can redo these with a lot of that data refined, so that we can build on our preliminary studies that suggest that this is an area that requires much further investigation. So in terms of a summary, I hope that you will agree with me based on the data that CpG is a very potent adjuvant. I hope that you will agree that biodegradable microparticles enhance the efficacy of CpG, that co-delivery of antigens and CpG generate robust antigen-specific immune responses, and that early evidence suggests that copper oxide nanoparticle inhalation, and not just copper oxide, but any toxic metal oxide, might interfere with vaccine efficacy and stimulate inflammation. And so the next steps in terms of where we need to go are to really look at dose responses, both for copper oxide nanoparticles by themselves, and those are ongoing studies that Sudhatip and Andrea have been sending me results even as I've been visiting here. And then looking at then the finesse combination or the fine-tuned combination of both, and then repeating those studies and then looking at dose response effects. And that's kind of the direction that we're currently heading in. We also want to do more studies looking at the immunological response in terms of subsets of immune cells based on exposure to the copper oxide nanoparticles with and without the dust mite allergy vaccine. And so to give credit to the people that are really my collaborators and the people that really drive this project, there's Andrea, who's an outstanding research scientist, my longtime friend and collaborator, Peter Thorne, Sudhatip, who's a graduate student in my group that is working together with Andrea. There's also Patrick O'Shaughnessy, who works together in our UO1 grant as one of the lead investigators. Ben Steins, Yifang, Ralph are all members of the consortium. Brittany Givens, who does a lot of the in vitro toxicity assays. And then to acknowledge Phil for the materials that he is sending to us, that we're really high quality materials, well characterized, that are helping us to do the studies that we're doing. And then I have an opportunity to ask you for questions and discussions. And before I jump into that, I'll go one slide ahead and tell you that the data that Andrea and Sudhatip have been emailing me while I've been here is based on this dose response trial where we're optimizing the amount of vaccine that we're giving, and we're doing something similar with the copper oxides, and we're looking at lung histology, inflammation, the extracts, the number of cells. And once we have gathered this full set of data and we've gathered all of the data on the copper oxide, we're going to combine both of those to have another study that takes this information to really look at the optimum conditions and to really see if we can develop a model that could be used for all of the materials that the ERCC is sending us that have the potential to stimulate inflammatory responses and also to mitigate vaccine-based responses. And so that's an ongoing project at the moment.